Hello and welcome everyone to today's live class session on the course Experimental Biochemistry, which is being taught by Professor Shagata Das Gupta and Professor Shomode of IIT Kharagpur. I am Srijan Sen, and today I will be taking your week 3 live session. So, I first of all I would be discussing with you the lecture in brief details, and then I would be discussing the assignments of previous years and a few questions that will help you in solving your assignments for this particular week 3 assignments, and also it would help you in taking up future exams. So there are going to be few questions from these previous years as well as from the assignments that you have been given to solve. So, okay, so let us start with our lecture slides. I am sharing the screen. Okay. I believe my screen is visible to you all. Okay, so let us start with today's lecture. So today's lecture will mostly deal with the spectroscopic techniques, where we would be discussing about UV visible spectroscopy, IR spectroscopy. Then we would be solving a few questions that will deal in these molar extinction coefficient, absorbance of say amino acids. Then we will be discussing in our future weeks about proteins and all. So let us start from the very basics of spectroscopy. So as we all know about spectroscopy, or we have heard of this term spectroscopy, that is the interaction of radiation with a particular sample. Okay, so interaction of radiation with a particular sample, the study of molecular or atomic structure of a substance by observation of its interaction with electromagnetic radiation. So when we speak of spectroscopy on in the perspective of quantitative means, it is for use for the determining the amount of material in the sample, and qualitatively it indicates the identification of the chemical structure of the sample. So as most of us are aware of many different ways of transmitting energy, and this phenomena comes together in one physical entity called as the electromagnetic spectrum. So there are many ways by which energy can be transmitted, and this particular set falls under the spectrum, the electromagnetic spectrum, where it all lies. And then there is the interaction of radiation with that particular sample. So there are rays that is being passed, and there is an electromagnetic radiation that excites that particular material in the sample. Okay. So the difference between these sources of radiation is the amount of energy that they radiate, and the radiation from these and other sources covers a range of energies. Okay, so these electromagnetic spectrum helps to understand the difference between the sources of radiation, that is the amount of energy that they are radiating, and all of these energy that they radiate uh, all comes under this electromagnetic spectrum, and this electromagnetic spectrum contains a huge range of energies. Okay. Now this is the electromagnetic spectrum. So if I speak about a few um, a nice information about say the history of your spectroscopy. So about, if we speak about the history of the spectroscopy, so it began during it began very early in your 17th century. Okay, so the modern spectroscopy in your started in the Western world in the 17th century. So it began with the design in your prisms, that is how we know as the optics, and it enabled systematic observations of your solar spectrum. So Isaac Newton first applied the word spectrum to describe the rainbow of colors, that is the splitting of light that combines to form your white light. And during your early 18 or during the early 19th century, Joseph von Fraunhofer conducted experiments with dispersive spectrometers that enabled spectroscopy to become a more precise and quantitative scientific technique. And these spectroscopy is even used in or played very significant roles in your astronomy, then physics and chemistry of that times. So Fraunhofer observed and measured the dark lines in the sun spectrum, which now bears his name, and this was also observed earlier by Wollaston. Okay. So all of these began in the 17th century and is still a very important thing to keep in mind. Okay, so the electromagnetic spectrum starts from the left hand side with the radio waves. Then we have the microwave, infrared. In this region we have visible light, UV, ultraviolet, X-rays, and the gamma rays. So towards the extreme left hand side, it is having a long wavelength, that is a lower energy and a lower frequency. So we know E equals to, or energy equals to, h nu. So energy is directly proportional to the uh, frequency. Okay, so that is why, and we know that nu equals to hc by lambda, so it is inversely proportional to your lambda, where c is the speed of light. So as the wavelength increases, the energy decreases alongside the frequency decreases, whereas gamma rays present at the extreme right hand side has the shorter wavelength and it has higher energy and ultimately higher frequency. So here what we need to keep in mind is the position of these waves or these rays in the electromagnetic spectrum, and which rays have the highest wavelength or the shortest wavelength and the highest energy and frequency compared to the lower energy and frequency. And the particular formula, that is E equals to hc by lambda, or E equals to h nu. So coming to the next important term, that is the radiation energy. So the strength of the radiation energy will interact with the molecules in different ways. So the energy that is radiated is the radiation energy. So high energy sources produce breaking of bonds like X-rays and gamma rays. So this is a very important slide because we need to keep in mind about the particular rays that is involved in the different function. So X-rays and these gamma rays are involved in breaking of bonds. UV visible spectroscopy excites electrons. The low energy sources produce vibrations in the chemical bonds. So vibrations in the chemical bonds is caused by infrared energy, and very low energy sources produce rotation of chemical bonds. So the rotation is done by the radio waves and the microwaves. Okay, so another time I'm repeating it. So for breaking of bonds we need X-rays and gamma rays. For exciting electrons, UV and visible spectroscopy. We have vibrations in chemical bonds by the infrared energy and rotation of chemical bonds by microwaves and the radio waves. Now, visible spectroscopy. So before we jump into visible spectroscopy, we need to know what is color. So we all know what is color. So color is a sensation which occurs when light enters the eye and it focuses on the retina at back of the eye. So it is the retina. So the light is actually initiates photochemical reaction in the nerve cells to the retina, and these transmit impulses into the brain and stimulate the sense of color. So it's observed over here, it's experienced in the walls of the retina, and it generates a photochemical reaction in the nerve cells, and that transmits signals into the brain, and that tells us what color is actually we observe. So all the colors that we actually sense are made up of three colors together with white, gray and black. Now there are two types of cells that we know, cones and rods. So cones gives color and three types which pick up red, blue and green, and the rods gives us gray, black, and also it is used for the night vision. So that is visible spectroscopy from the perspective of eye or color. So what is the composition of the white light? So sunlight is the white light that covers a wavelength range of 350 to 750 nanometers. Now if we carry out, we have all performed this physics experiment. Okay, so it shows that white light is actually a composition of a range of colors, light with different energies and different wavelengths. So when we put a white light on a prism, it splits into 7 different colors. So the less splitting is of red light, and the most splitting is what we observe for a violet light. We have all done this in our school, while working with a prism and putting a white light on it. So when white light falls on an object, the color detected by the eye will depend upon the absorption, reflection properties of that material in the object. Now if we observe a particular object to be appearing white, that means it has reflected all the colors, and if the material completely absorbs all the light, it appears to be black. Now if the material absorbs a constant fraction of light across the spectrum, it appears gray. So three different colors have been justified, the two extremes being white and black. So if it reflects all the light, it is white. If it absorbs all the light, it is black. And if a partly constant fraction of light is absorbed, then it appears to us as gray. So these are the different wavelengths of light that is absorbed, and the color absorbed is what we tell is a very contradictory color to what is seen by the eye. So what is absorbed, so the color that is reflected, okay, so the color that that material or that object reflects is what our eye captures. So it is very different from the color that is absorbed. So for example, if the object absorbs color violet, then we see it as yellow green. If it absorbs blue, we see it as yellow. If it absorbs green, we see it as purple. If it absorbs orange, we see it as green blue, and if it absorbs red, we see it as blue green. So when a sample only absorbs light of a single wavelength, the eye sees the complementary color. So and the wavelengths of the visible lights have been put over here, so you can check that. Like violet has a wavelength range of 380 to 430, blue has 430 to 480, green has 500 to 560, yellow has 560 to 590, orange 590 to 610 nanometer, and red has 610 to 750 nanometer. So there is a wide range of visible spectroscopy which starts with 380 nanometer, and in the end we have the 750 nanometer. So this visible spectroscopy lies within 380 to 750 nanometer, and what the object reflects is what our eye absorbs. Okay, so we observe the complementary color. So these are the different effect of energy levels depending on the nature of the energy that we receive. So if there is an absorption by UV visible spectroscopy, so it moves from the S0 or the ground state to the rotational level, and when there is an IR spectroscopy, it moves from the ground state to the vibrational levels, and when there is a microwave, it moves to the rotation levels of that particular energy. So This is the ground state and this is the excited state. So UV visible has a very high. So if we go over here, so what happens with the UV visible? So it has a high energy, whereas radio waves have low energy, infrareds have less energy. Okay, so greater the energy, greater will be the absorption, and this UV visible ray is enough to excite that particular molecule from the ground state to the S1 excited state. Next, when there is an infrared, it moves to the vibrational levels of that particular state. So a state has rotational levels and the vibrational levels. Okay, so there is a, for each state, like we have S0, S1, S2, say Sn. So each state has rotational levels and the vibrational levels. So microwaves moves from S0 ground state to the S0 rotational levels. Okay, from IR from S0 ground state to the vibrational levels of that particular S0 state. And for UV visible, it moves from the rotational level of the ground state to the excited state. So higher the energy, higher will be the excitation. So UV radiation has a wavelength range of 220 to 380 nanometer and the visible radiation between 380 to 780 or 750 nanometer. So substances can absorb varying amounts of UV or visible radiation at particular wavelength. So colored compounds absorb energy in both UV visible region of the electromagnetic spectrum, and substances can be liquids or solids, and measurements are made with instruments called spectrophotometers. So modern instruments can be coupled to microscopes, which allow solid samples and very small samples of solids and liquids to be analyzed both qualitatively and quantitatively. So here we have a UV visible spectroscopy where the incident light is passed at a wavelength of 254 nanometer through a particular sample having intensity I0, whereas the transmitted light that we get is IT. So we have the intensity that was passed was I0 and the intensity that we receive is IT. So if a particular wavelength of UV or visible radiation can be isolated from the source and pass through the sample, which can absorb some radiation, then the transmitted light intensity is IT, which will be less than the incident light I0. So where is the left out amount of incident intensity gone? That is absorbed by the sample. So the amount of light transmitted with respect to the incident light is called transmittance. So T is the transmitted, that is the amount of light transmitted with respect to the incident light. So T is equal to IT by I0. Now if we need to calculate transmittance in terms of percentage, it is IT by I0 into 100. Now the question is, that is transmittance. So what we want to know is mostly about what has been absorbed. So there is a very nice relationship between absorbance and transmittance, although they are not directly linked. So how are they linked? So before that we need to, because what we are seeing, that is what we have to put it in a data. Like we do not see observe the absorbance over here, we do not get the absorbance directly. What we have in hand is the incident light intensity and the received light intensity or the transmitted light intensity, that is I0 and IT. So transmitted light is IT by I0 into 100. Now absorbance is equal to, so absorbance is equal to A equals to minus log to the base 10 of transmittance, where T equals to IT by I0. I have shown it over here. Okay, and these equations are very important because you need to keep in mind, because there will be many questions in your assignments or your competitive exams as well as your exam. There is going to be short questions from these portions. Okay, so absorbance is equal to minus log to the base 10 of transmittance, and transmittance equals to transmitted light intensity divided by incident light intensity, that is IT by I0. So absorbance is equal to log to the base 10 of I0 by IT. So the negative term is taken inside, it is I0 by IT. For the percentage transmittance to be 0 and 100, the corresponding observance values would be 0 and 2. So that is fine. So how do we get absorbance? So if we plot absorbance versus wavelength, we get an absorbance spectrum is generated for compounds of A and B. Now with the advantage that absorbance measurements are usually linear with concentration, absorbance spectra are now used. So we plot absorbance versus concentration and we get a curve like this. So this is absorbance and this is plotted against concentration. Okay. Now before we get into the main equation of absorbance that we will be needing for different solving different questions or for finding out the molar extinction coefficient of that particular compound or that solution, we need to keep in mind very important laws of spectrometry. So there are two very important and basic laws that we need to keep in mind. One is the Lambert's law and the other is the Beer's law. And the third one is the combination of the two, that is Beer-Lambert's law, or sometimes it's written as Lambert-Beer's law. Okay. So what do these two laws tell? So Lambert's law speaks about absorbance being proportional to the path length of the absorbing medium. So absorbance proportional to L. Whereas Beer's law tells absorbance is proportional to the concentration. So A proportional to L and here A proportional to C. So if we combine the two, absorbance is proportional to C and L. So therefore it is substituted, so it's proportional, so we need a proportionality constant. So absorbance is A is absorbance, C is concentration, L is path length. Then we introduce a proportionality constant, that is epsilon. So epsilon is the molar extinction coefficient. So what can be the molar extinction coefficient? It can be moles per liter. Path length is concentration is moles per liter. Path length is centimeter. So if we get mole inverse liter centimeter inverse. Also, it can be understood as 1000 mole inverse centimeter, 1000 centimeter square mole inverse. So it has two concentrations. One is 1000 centimeter square mole inverse and the other is mole inverse liter centimeter inverse. Now the importance of Lambert-Beer's law is that it simplifies this particular graph where we are plotting absorbance versus wavelength and we get a lot of curves and all, but this is not a very nice, it does not have a very direct relationship with it. So when we get a very linear relationship, it actually makes the calculations a lot more easier. So we have got A equals to epsilon Cl. So if E and L are constant, absorbance is directly proportional to concentration and it should have a linear relationship. So what we need to do is we need to prepare some standards of the analyte to be quantified at known concentrations and measure the absorbance at specific wavelength. So the wavelength is constant, kept constant over here. Then we prepare a calibration curve for then from measuring absorbance of the sample. So the concentration of the analyte in the sample can be obtained from the calibration curve. Epsilon can be obtained from the slope of the calibration curve for a given wavelength. So here we are plotting absorbance versus concentration, and then we get at a particular concentration we get the absorbance. And this is the you call as the calibration curve. So these are known standards, and you know for knowing the concentration you calculate the absorbance where you know the value of the epsilon. If you know the molar extinction coefficient, what you can directly do is you put it, you get the absorbance, and from there you get the concentration value. In this manner you can get a linear relationship and that will help you. Next, we would be talking about the Jablonski diagram, but till now, over here, we have spoken about the spectroscopy. Okay, so spectroscopy, the electromagnetic spectrum, radiation energy, visible spectroscopy. We saw the effect of the energy levels on the different nature of the energy that is received. We spoke about the UV visible spectroscopy, the laws of spectrometry, and the Lambert's-Beer's law. Next, we would be speaking about the Jablonski diagram in a few minutes. Hello.
Good evening, sir.
Good evening.
I'm trying to very long time, but I'm not getting.
Okay, it's not a problem. Fine. Um, my slide is visible, right? Is my slide visible to you?
Not sir. Now getting.
Okay. Now it's visible, right? Okay. So just I will give a brief. I will just give a brief idea on what I discussed. Okay. So first we spoke about
Uh,
spectroscopy, where these are just the basics of spectroscopy, where we talked about the electromagnetic spectrum, and we have the radio waves with the longer
uh,
wavelength and the gamma rays with the shorter wavelength, and we have a very nice formula, an equation where we have H equals to energy equals to hc by lambda, where lambda is the wavelength and c is the speed of
uh,
light, and the other one being h equals to e- equals to h by h lambda, h nu. So nu is the frequency. So higher the energy, higher will be the frequency, and higher the energy, lower will be the wavelength. So in this way we have segregated the different rays. Then we spoke about the radiation energy, where we spoke about the breaking of bonds being produced by X-rays and gamma rays. We excite electrons by UV visible spectroscopy. We produce vibration in chemical bonds by infrared energy, and the rotation of chemical bonds by microwaves and radio waves. Then this is just a basic of the visible spectroscopy, where we use the eye as the optical instrument for observing the color, and the color that we observe is complementary to the color that is being absorbed by the sample or that particular material. So here the color that is absorbed by the material is the color, the complementary color that we are actually observing. So this is the wavelength of range absorbed. Okay, and then we have the effect of the energy levels, where higher, where when we are providing UV visible light that is of higher energy, it is moving from the ground state to the another higher orbital. So each levels have rotational as well as vibrational levels inside them. So S0 being the ground state has rotational and vibrational levels, and S1 being a higher energy state has rotational and vibrational levels. So UV visible light excites it from the ground state rotational level to the S1 rotational level, whereas IR from rotational to the vibrational level of S0, and microwaves moves it vary from the rotational level 1 to the rotational level say 4. So we talked about this. Then we spoke about the UV visible spectroscopy. So UV has a range from 220 to 380 nanometer, and visible radiation from 380 to 750 nanometer. And substances can be your liquid, solids, can be anything, but the measurements are made with instruments called spectrophotometers or the spectrometers. Next, we talked about the most important equations like We have transmittance. So a particular incident light is passed to the sample, and this is the transmitted light. I0 is the incident light, IT is the transmitted light. So it can be equated as T equals to IT by I0. And then transmittance can be calculated as percentage, where percentage transmittance equals to this. Now this is transmittance, but what we need to get is absorbance. So absorbance can be equated as A equals to minus log to the base 10 of transmittance, and then when we calculate it, we get this kind of a curve where we have absorbance versus, this is absorbance and this is your wavelength in nanometer. So we get a curve like this, but it is very difficult to actually uh, claim anything or conclude anything from this graph about the absorbance. So for this we came across the two important laws of spectrometry, the Lambert-Beer's law, where absorbance is directly proportional to concentration and path length, and for this we have a proportionality constant, which is known as the molar extinction coefficient, and there are two units of molar extinction coefficient is mole inverse liter centimeter inverse and 1000 centimeter square mole inverse. And then the importance of Lambert-Beer's law is that we can actually equate this particular curve. The curve that was difficult to calculate because of its complexity, it becomes easier when we get a linear relationship. So these are the standard points that has been plotted, and just when we know the absorbance of that particular value, we can know the concentration. So from this we get to know about the concentration of the particular sample. Now what we will be talking about is the Jablonski diagram. So for this we have very important terms of absorption, fluorescence and phosphorescence. So these are very important terms when we speak of spectrometry. But in brief, if I tell, so absorption is movement, is excitation from the ground state to a higher state. That can be your S2, S3 or S1, whereas fluorescence is the, so absorption and these two are actually emissions. So this is absorption, so absorption from S0 to the S1 state and the S2 state. So if I show over here, so it's this is the ground state, this is the S1 state, that is the singlet state and the singlet 2 state, whereas over here T stands for triplet 1 and triplet 2. So these are the different levels. So absorption is excitation from the ground state to the higher orbitals, whereas as fluorescence is indicated by, so here when the S2, so fluorescence is emission from your higher molecular orbital to the lower state, that is, but within the same states. Like for the singlet, it is singlet only. For phosphorescence, it is from the triplet to the singlet state. Like over here it comes from the S1 state to the ground state. So fluorescence, there is an emission from the S1 state and the electrons comes down to the ground state. For phosphorescence, it is from the T1 state, that is the triplet state, to the ground state. Now how do they change this different levels or the different states? Yes, for this there is an absorption that is taking place, and then there is an internal conversion where the electron moves from the S2 to the S1 state. So this conversion is known as the internal conversion or the loss of energy. Now when the electron is in the S1 state, it moves to the ground state via fluorescence. So that is another internal conversion, or IC. Now inter-system crossing is when the singlet state electrons comes to the triplet state of the same level from S1 to T1. So when there is a system that is changing, we call that as the inter-system crossing, or ISC. So now from the triplet state, when it goes down to the ground state, we call that as the phosphorescence. So S is the singlet state and T is the triplet state. Okay. So another time I'm repeating, it's a very important thing that we need to keep in mind. So absorption is when the electrons are excited from the ground state to the higher molecular orbitals or the higher molecular states, can be S1, S2 or Sn. Now there are two terms for emission. One is fluorescence, one is phosphorescence. So fluorescence is when there is an intersystem, when there is an internal conversion of that electron, where the electron loses energy and comes down to the from a higher level to a lower level. We call that as the internal conversion. Now from the S1 state it goes down to the is 0, but there is no system that is changing, that is singlet is remaining in the singlet, but there is a loss of energy or an energy is emitted. So that is fluorescence. But phosphorescence is a change of system but of the same level. So change of system from singlet to triplet, and the same level, that is 1 remains 1. So S1 to T1, and then phosphorescence, emission of energy, and the electron comes down to the S0 or the ground state. So here we have the different molecular orbitals. So starting with from the ground state we have sigma, then we have pi, and non-bonding orbitals, then pi star and the sigma star. So non-bonding to pi star is less than pi to pi star. Then these are the different energy levels. Okay, then sigma to pi star, then we have pi to pi star. So higher energy for pi to pi star, then we have pi to sigma to sigma star is the highest energy, then sigma to pi star, then we have non-bonding to pi star, then we have pi to pi star, non-bonding to sigma star, pi to pi star, and non-bonding to pi star. So this is the lowest energy, and these are given over here. Okay. Now the next term is Stokes shift. So Stokes shift is the gap between the maximum of the first absorption band and the maximum of the fluorescence spectrum. Okay, so these are the two maximas of absorption and emission. So your Stokes shift is the wave number of absorption minus the wave number of emission or fluorescence, and that is indicated by a delta of the wave number. So that is Stokes shift, the difference between the first absorption band and the first fluorescence spectrum. So loss of vibrational energy in the excited state is as heat of collision with the solvent. So any energy level the electrons are in, it will come down to the V0 or the ground state of that particular level, and that loss of energy in the excited state is by the collision with the solvent.
गलत है
Yes.
हाँ
Okay. Now these are the examples of different fluorophores that we have over here. So one is your fluorescein, and the other one is ethidium bromide bound to the DNA. So for this, what we have is, so we have fluorescein that has an absorption at around um, say what do I see 480, and it emits at around say 520. Whereas for ethidium bromide bound to DNA, so we all know that ethidium bromide intercalates within the strands of your DNA and it fluoresces. So it has an absorbance at around 520 nanometer and it emits at say 625 nanometer. So the next one is the lifetime. So excited states decay exponentially with time. So minus I is equal to I0 e to the power minus t by tau, where I0 equals to initial intensity. I is the intensity at after time t. Tau is the lifetime of the excited state, and kf is the constant factor for fluorescence, that is 1 by tau. So sometimes it can be written as e to the power minus t kf, because 1 by tau equals to kf. Okay. The next very important term is quantum yield. So quantum yield is the number of fluorescence quanta that is emitted divided by the number of quanta absorbed to a singlet excited state. So quantum yield is the ratio of the photons emitted to the photons absorbed. So in simple words, it is the ratio of photons emitted to the photons absorbed by the system. So fluorescence quantum yield is this one is fluorescence quantum yield. So we have photons emitted by photons absorbed. Then we have the fluorescence lifetime and the radiative fluorescence lifetime. And these are complicated equations of these quantum yield, where we have k equals to all the summation of all these terms of we have the fluorescence radiative rate, then we have the internal conversion, that is by the collision. um, Collision with the solvent plus the internal vibrational modes that increases with your temperature. Then we have the quenching constant that is obtained by the collision with solute molecules like such as O2. Then we have the energy transfer by FRET, then reaction from the excited state and the intersystem crossing factors. So all of these adds to give us this k. Now where do we need this k? So these are the photons absorbed. So photons can be absorbed by all of these means, and that is the denominator. The numerator has the photons emitted, that is kr. So what is kr? So kr is the fluorescence radiative rate. So it is photons emitted by the photons absorbed. Now polarization. So molecule of interest is randomly oriented in the rigid matrix, and plane polarized light is used as the excitation source. So the degree of polarization is defined as P. So your degree of polarization, or P, is equal to your intensity at the parallel component minus the intensity at the perpendicular component divided by the addition of the two intensities observed by parallel and the perpendicular components. And alpha is the angle between the emission and the absorption transition moments. And if alpha is 90 degrees, then P equals to minus 1 by 3, and if alpha is 0 degrees, then P is equal to plus half. Why? Because it is equated in the cos function, where polarization is equal to 3 cos square alpha minus 1 divided by cos square alpha plus 3. Okay. So that is the angle, alpha is the angle between the emission and the absorption transition moments. Now the other thing is the inner filter effect. So at low concentration, the emission of light is uniform from the front to the back of the sample cuvette, and at high concentration, more light is emitted from the front than the back. Since emitted light only from the middle of the cuvette is detected, the concentration must be low to assure accurate measurements. So we need to use low concentration, because it then the emission will be uniform. Whereas for high concentration, what happens is that it is mostly emitted from the front than the back of the cuvette, so the emission becomes the accuracy of that measurement becomes very uncertain. So we know that different amino acids absorb at different wavelengths and they have different absorptivity. So tryptophan absorbs at 280, tyrosine 274, and phenylalanine 257. And the absorbability of these is 5600 for tyrosine, 1400, and for phenylalanine 200. So we have all of these, and these play a very important role in the intrinsic fluorescence of your intrinsic fluorescence of proteins and peptides. Okay. So we would be speaking about the absorbance when also we will be speaking about proteins, and previously we spoke about this when we also talked about peptides and amino acids. Okay. So but this much is actually required for your the spectroscopy class. So next we would be discussing a few practice problems. Okay, so we would be talking about the practice problems in say
Hello, sir.
hello.
I'm waiting for you. Okay.
Hello,
You have taken a break?
can you switch off your camera, because otherwise the connection is getting disturbed because I
Ah,
have low connectivity. So yes, good. Yeah.
ah,
Oh no, actually my network
I,
connection got disrupted and I was actually kicked out of the meet. Okay. Yes, I'm sharing my
ah,
slides once again.
sir, about in assignment third questions regarding that
About
also explain.
today's assignment.
Assignment number three.
Yes, if you have questions, you can ask me. You can ask me. Do you have a
Um,
question? You can ask me now.
that assignment copy I have not yet. So it is difficult to be at the same time open the questions and ask, because I am
Okay, so
using
what you
mobile
can
very
do,
fast.
okay, what you can do is you can mail me the question in this mail ID. I would be getting back to you. Okay, so anytime you face a problem, that is applicable for anyone actually. So you can mail me
Yes,
over here. I will actually answer the question over there only. So that will be easy. Okay, you can just mail me the question.
now once again, sir, show me your email address.
Sorry,
So I take that screen.
sorry.
I will take a screenshot your email address.
Yeah, yeah, no, I have just put it in the chat, so you can just check it. Okay.
Uh,
So you get back to me means you mail me. I would get back to you over mail only, right?
uh,
Okay, good. Okay, now let us discuss with today's practice problems. So the first question says the region of electromagnetic spectrum which has a wavelength of zero point zero 0.01 to 10 nanometer. So what do you think would be the answer? So wavelength of zero point zero 0.01 to 10 nanometer. Okay, let us go back to where we talked about this. I don't think we would be getting it over here, but let us see. Do we have an answer to that? So zero point 0.1 to 10 nanometer. So wavelength is shorter than this. So it can be either UV, X-ray or gamma rays. Let us see what are the options. So we have X-ray, infrared, UV and the radio wave. So if you see, we do not have over here, but if you go to the electromagnetic spectrum of all these particular rays, you will know that the correct answer would be the X-ray region. So A is the correct option. Now the next question is, if the frequency of the radiation is increased by 2 times, if the frequency of radiation is increased by 2 times, what would be the change in the energy of radiation? So frequency is increased by 2 times, how would the energy change? So to solve this, we have that particular equation where E equals to H nu, and nu is the frequency. Okay, so E equals to H nu, so it increased 2 times means the frequency will also increase 2 times. So B is the correct option. The next one. The question says the type of radiation that often triggers molecular vibration. Okay, so molecular vibrations are triggered by. So if you can remember that when I was speaking about that the different rays that is being used at different for exciting the uses of different radiation energies. So for breaking of bonds, we have X-rays and gamma rays. For exciting electrons, we have UV visible spectroscopy. And for vibration of chemical bonds, we have the infrared energy, and rotation of chemical bonds, we have microwaves and the radio waves. So for vibration of chemical bonds, we have the infrared energy. So let us see what are the options. So we have infrared radiation, we have microwave radiation, we have UV radiation and the X-ray. So 2 is 1, 2, 4. No, 1 is not correct. 2 and 4 will be correct, where we have microwave radiation and the infrared radiation. The next question is, an electron is excited from non-bonding orbital to a pi orbital. The correct option is, okay, this is an easy question. So for non-bonding orbital we represent it by n, and for pi orbital it is represented by pi, and antibonding, so star. So star indicates the antibonding orbital, and for that particular orbital, so if it is sigma star, it is um, sigma orbital, but it is anti-bonding. If it is only sigma, it is bonding orbital. So it says from non-bonding to pi star, pi anti-bonding, so pi star, so into pi star. So this will be the correct option, C. Coming to the next question. In the above graph, what kind of shift do you observe in B with respect to A? So with respect to A, what shift do we observe in B? So this is A. So what is happening? So two things we need to observe. One is that it has shifted to a shorter wavelength, and number two, it has the absorbance has increased and it has shifted to a shorter wavelength. So let us discuss what are these all of these shifts. So what is hypsochromic shift? So hypsochromic shift is a shift to a shorter wavelength. Now bathochromic shift is shift to longer wavelength. Hyperchromic shift increased absorbance, and hypochromic shift is decreased absorbance. Okay, so all of these are the meanings of all of these shifts. So now when we see is that observance in B with respect to A. So the absorbance has increased, so hyperchromic, and it has shifted to a shorter wavelength, so hypsochromic. So hypsochromic and hyperchromic. So 1 and 3 are correct. So B is the correct option. Do you have any question?
No, sir.
Okay, good. Good to go then. Okay, the next one. Now, UV absorbance at 280 nanometer is caused by tryptophan residue. The absorbance is 0.35, and for 1 millimeter path length, and we have been given the epsilon value. So let us try this calculation. So we know this equation. So E is given as 1000, C is not known to us, L is given as 0.1 centimeter, and we know absorbance is 0.35. So E is equal to 0.35 divided by 1000 into 1. So what do we get? So we get it as, sorry, this would be C. So we get it as equal to, so over here 1 and 1, so 4. So we get it as 3.5 into 10 to the power minus 4 molar. Right, so 10 to the power minus 4 molar, that is equal to your 0.35 millimolar. Okay, so that is 0.35 millimolar. So we get it as this one. Okay, so D is the correct option. A equals to epsilon C, all the values are given, so we need to calculate just C. So C equals to 3.5 into 10 to the power minus 4 molar, so 0.35 millimolar. The next one. Okay. The next question is, a student is going to measure the concentration of RNA protein in distilled water. What types of solution would he use to conduct the experiment in cuvette A and B? So A has RNase A and B has distilled water. C1 C2 equals to RNase A distilled water, B is RNase A, and not enough information is provided. So if you see that only what has been mentioned is that a student is measuring the concentration of RNase A in distilled water. So what type of solution would he use to conduct the experiments? So nothing has been spoken about the cuvettes or what has been there in the cuvettes. So we would be concluding over here as not enough information has been provided. Nothing else to deal with it. Okay. And that is not even a very important or an exciting question, so it's fine enough. Okay. The next one. The double beam, okay, let us solve this one first. This amino acid absorbs UV region mainly due to, okay, so the absorbance of the amino acid is mainly due to what? So the options given is COOH, NH2, then we have the OH group or the phenolic group. So if we think of this amino acid, it is your tyrosine amino acid. Okay, so we have the COOH group, NH2 group, and the OH group. So we know that for tyrosine, for tryptophan, and the phenylalanine, the phenolic group, or not only the phenolic group, the benzene ring is very important. Okay, so that is why for this tryptophan residue, the tyrosine residue, the phenolic group is playing a role in the absorbance. So this group is important. The benzene group or the aromatic groups that are present plays an important role. The next one, the double beam spectrometer differs from the single beam spectrometer. So for this we have a very nice diagram that we can understand over here. So how does this double beam spectrometer differ from a single beam spectrometer? So there is a light source, then there is a double beam spectrometer. So we have the sample beam that passes through the flame and it goes into the monochromator and the detector. But the other beam that goes to the as the reference beam, and that mixes over here and goes to the monochromator, and we have the detector. So both the sample and the reference cuvettes cannot be measured simultaneously. Reference cell lamp has a different wavelength from the sample cell lamp. Chopper splits the light into two parts, which passes through the reference and the sample cuvette, and reference absorption is first recorded, followed by the sample absorption. So what happens, both the samples cannot be measured simultaneously. That is incorrect, because it can be measured simultaneously. Has a different wavelength from the sample cell length. That is also incorrect. What is correct is that the chopper splits the light into two parts. So we have a chopper over here that splits it into two parts. One is the sample beam and the other is the reference beam. So the double beam spectrometer differs from the single beam spectrometer is that we have a chopper that splits the light into two parts. Okay. The next question. Which of the following statement is true for UV visible spectroscopy? So an absorption band at 190 nanometer is due to disulfide bridges between two cysteine residues. The cuvettes are made of quartz with two sides frost and the other two sides transparent. The peptide bonds are chromophore with strong absorption band at 280 nanometer. The weak absorption band for the peptide bond comes around 190 nanometer. So which of the following statement is true? For disulfide bridges, the two cysteine bonds absorbed at 190 nanometer is incorrect, and the weak absorption band for the peptide bond comes around 190 nanometer is also incorrect. And the peptide bonds are chromophores with strong absorption bands at 280 nanometer. That is also incorrect. So peptide bonds absorb at 220 nanometer, not 280. So the correct option is B, where we have the cuvettes are made of quartz with two sides frost and the other two sides are transparent. So option B is correct. Now the next one. A researcher has prepared a new drug molecule to study interaction with the protein. The lambda max, or the maximum wavelength of the drug molecule, is 260 nanometer. So the desired emission wavelength must be. So what we are seeing over here is that a researcher has prepared a molecule to study the interaction with the protein. The lambda max of the drug molecule is at 260. So where will be the desired emission be? So for this we need to keep one thing in mind, is that the emission should be at least 10 to 15 nanometer ahead of the excitation wavelength. Okay, so the gap has to be about 10 to 15. Okay, so if we see over here, the correct option would be most probably be 275 nanometer. Because why is it so? So the emission should be at least 10 to 15 nanometer ahead of the excitation wavelength, because in Jablonski diagram, if we see that the molecule once excited to a higher molecular orbital, say S2 from S0, it comes down to the lowest vibrational level of S1. Now the energy gap between S1 to S0 is lower than S0 to S2. So hence the fluorescence wavelength is in a higher range than the excitation wavelength. So E is directly proportional to, or inversely proportional to 1 by lambda. So much higher wavelength should be avoided, since 1 mem is the emission maximum. So for that it has to be at a higher, but between 10 to 15 nanometers. So 10 to 15, so 275 nanometer is the correct option. Okay. The next one, which of the following statement is or are true? Monochromators are used to disperse white light into the various wavelengths. At low concentration, high emitted from all parts of the fluorescence quartz cuvette can be considered to be almost uniform. Fluorophore and chromophore are same thing, and the maximum excitation wavelength for tryptophan, tyrosine and phenylalanine is 258, 275 and 280 nanometer respectively. So we know that the last sentence is incorrect. The last sentence is basically incorrect. So what we have over here is the next one. Fluorophore and chromophore are the same thing. No, they are not the same thing. Fluorophore are fluorescent chemical compound that can re-emit light upon light excitation, and chromophore are or chromophore is an atom or group whose presence is responsible for the color of a compound. So one is responsible for color of a compound, another one is responsible for the fluorescence. So those two are different. So monochromators are used to disperse white light into various wavelengths. That is also incorrect because it's after all a monochromator, so it splits into a particular light. And what is correct? So monochromators are used to disperse white light into various wavelengths. That is correct. And at low concentration, the light emitted from all parts of the fluorescence can be considered to be almost uniform. That is also correct. So 1 and 2 are correct, but the other two are wrong. Okay. The next one is another choose the correct option statement is in fluorimetry. Photovoltaic cell and PMT are used as detectors. When molecules are excited in visible region, hydrogen deuterium lamp is used. Compounds having aliphatic carbonyl structure shows more intense fluorescence than that of compounds having aromatic functional group, and quantum yield quantifies the efficiency luminescence. So when molecules are excited in the visible region, xenon lamp is used, and compounds having aromatic functional groups shows more intense fluorescence than that of compounds having aliphatic carbonyl groups. So option 3 is incorrect and option 2 is incorrect. So 1 and 4 would be correct. So B is the correct option. So whatever questions I am actually discussing is very important for your final exam on this course, okay? Because there would be few common questions or similar type of questions in your exams as well, and not only for exams. This course is also very beneficial for a different competitive exam. So solving the assignments or coming to this live session is very important. Okay, so I think we have one more question to go. Okay, oh, we have two more. Okay, so while taking fluorescence emission of lysozyme protein having phenylalanine, tyrosine, and tryptophan residues, one should select the excitation wavelength for. Okay, so one should select the excitation wavelength for tryptophan residue, since tyrosine and phenylalanine is non-fluorescent in lysozyme protein. Tyrosine, since tryptophan and phenylalanine is non-fluorescent in lysozyme protein. Tryptophan residue, since tyrosine and phenylalanine have much lower quantum yield as compared to tryptophan. And either tryptophan or tyrosine, since phenylalanine is non-fluorescent in lysozyme protein. Okay, so this question has to be answered from the perspective of quantum yield. So if you see that nothing has been said about the combination of amino acids in that particular protein lysozyme. So let's answer it from the quantum yield. So tryptophan residue, the excitation wavelength has to be selected for the tryptophan residue, since the quantum yield for tyrosine and phenylalanine is much lower than tryptophan. We have seen about the absorptivity. If I go to that slide once again, so you see over here, so we have the quantum yield of 0.2 for tryptophan, tyrosine has 0.14 and phenylalanine has 0.04. So even if you add these two, it is not close to 0.2. So that is why it is better to set for tryptophan than for the other two residues. And the last one is the correct path of light in a spectrofluorometer. So the correct path, let us see what is the correct path. So the first one, it is source lamp, sample chamber, fluorescence monochromator, detector, recorder. Okay, next one is recorder, monochromator, detector, sample chamber, and fluorescence monochromator. Then we have source lamp, monochromator, fluorescence monochromator, detector, sample chamber, and recorder. This is also incorrect. Recorder cannot start from the beginning. This is also incorrect. Source lamp, sample chamber. No, first it has to be source lamp, then monochromator. So source lamp, monochromator. Yes, this is correct. Then this monochromator will go into the sample chamber. Okay, yes. So the last option is correct. We have the source lamp, monochromator, sample chamber, fluorescence monochromator. Then we have the detector, and finally we have the recorder. So the last option is correct. So that is all for from the previous year questions, and also a few important questions were there. Now if you have any doubts, you can ask me. Any doubts or queries?
Actually, sir, I didn't study well. So, and apologize for this. I will study next
No,
time
no,
very well.
it's
Then I
fine.
will ask.
It's fine. Means you don't have to be sorry about that. It's absolutely fine. Sometimes you get busy, but the good thing about this is that these lectures are being recorded and will be uploaded in the YouTube very soon. And the NPTEL team would be
Uh,
sharing with you all of these recordings and the lecture slides. So you can definitely go back and see what has been taught. And also I have provided my email ID in the chat box, so you can definitely get back to me anytime when you are up with a query. Okay, so that's absolutely fine. That's why it's good that these video lectures are recorded, so if people are busy or students are not able to join the class, they can actually listen to these lectures. But also it is also
yes, yes.
important to join the classes for these live sessions and one-to-one discussion, because it will be beneficial to clear your doubts in one-to-one session, because the lectures that you go through for every week, that is also being recorded. So the live sessions are actually conducted for the betterment of the students and to get their doubts cleared face to face. Okay, but that's fine.
Definitely.
Sometimes it's you get busy with different work, and okay, so that's okay. It will be uploaded in YouTube. Any more doubts? If there are no more doubts, then we can end the class. So thank
Uh,
you. Sorry.
I, yes, sir. If you want to end the class, I am agree
Yeah.
with you.
Do you have any more doubts?
No, sir. Now, not now.
Okay. If you are having any doubts, you can get back to me in my mail. You can write to my mail. I will check it and answer you. Okay.
Yes.
Okay. So thank you all for joining. Thank you. So we end
Thank
the
you, sir.
class over here.
Okay.